Good evening, everyone. My name is Vishal Joshi, and I am a software engineer working with European Bioinformatics Institute in UniProt team. Today, I'm going to talk about CROssBAR API and its use in building knowledge graphs. Before we dig into CROssBAR API, we should briefly talk about what CROssBAR project is. CROssBAR stands for Comprehensive Resource of Biomedical Relations with Deep Learning Application and Knowledge Graph Representations. This project has been developed in collaboration with EBI and Middle East Technical University, Ankara, Turkey. There are several computational tools and services which have information which is highly scattered and lacks connectivity, thus making it highly unlikely to be used for real-world problems. CROssBAR aims to develop a comprehensive computational resource to address these shortcomings, which focus drug discovery and precision medicines. In addition to bringing the data together, it also predicts missing information using its deep learning data models, and the architecture is known as Deep Screen. Now, CROssBAR API. CROssBAR API is an open access web service, which means it allows public access to all of its data through HTTP endpoints. The API follows OpenAPI specification, which defines a standard language agnostic interface to RESTful APIs, which allows both humans and computer to discover and understand the capabilities of the service without access to source code, documentation, or through network inspection. The API also, in addition to being RESTful, it allows an interface which can see when you click on this link, which allows users to try out the web service and see the data yourself, formatted within the same browser without the need of a REST client. Finally, this API can be used this API provides a multifaceted view of stored data through 12 endpoints, which allows users to drive biomedical networks from any facet, for example, drug-centric, disease-centric, or gene-centric. As mentioned earlier, CROssBAR API endpoints, there are 12 CROssBAR API endpoints which have been developed using RESTful architecture. As you can see on the left-hand side, the CROssBAR data API Swagger documentation screenshot. The 12 endpoints are from activities to targets. If I briefly describe what these endpoints are, the first endpoint is Campbell, exposes the data from Campbell activities which has been stored in CROssBAR. The second one is Campbell assays stored in CROssBAR DB. Drugs are from drug bank stored in CROssBAR DB. EFO data in CROssBAR DB. HPO endpoint exposes the HPO data in CROssBAR DB. The Campbell molecules, the UniProt proteins stored in CROssBAR DB, the PubChem dataset, and the Campbell targets. If you see on the right hand side, these are the recent database statistics, the latest ones across all these data stores for all these data entities. So it starts from Campbell, stores activities, assays, molecules, and targets, which we have shown on the left-hand side. The UniProt proteins, drug bank, the PubChem, bioassays, compounds, substances, EFO diseases, and HPO phenotypes. Now coming to the graph construction part. This is how a graph can be constructed using CROssBAR API. This graph construction happened using manual steps, but now using CROssBAR API, these endpoints can be weaved together to develop a final knowledge graph, which will look like this. If you see how the graph construction works, the first step is for this example to represent a hepatocellular carcinoma knowledge graph is, first we gather the genes across all these databases. For all these genes, we query the database to find out the proteins related to these genes and the protein-protein interactions. After gathering the protein interactions, we find all the drugs which target the proteins collected in step 2. We also incorporate the predicted drug target instructions using our state-of-the-art deep screen deep learning algorithm. After gathering these compounds interacting with these related genes, we go for all the signaling pathways associated with STC disease pathways. And the final step is to determination of the other diseases and phenotypes which are related to the final set of genes which we have identified after the first four steps. If we combine the data, it can be represented into this knowledge graph. It's a highly complex graph, but it has been developed with step-by-step procedure to come up with this highly complex but highly representative information. The red nodes represent the diseases, the purple nodes are the phenotypes, the signaling pathways are in blue nodes, and the drugs are in red. The compounds, these are the compounds, and the greens are genes and proteins. We utilize this phenomena of using CROssBAR API for biomedical graph construction for COVID-19 knowledge graphs. As you can see, we used the UniProt database to find out the organisms which form a part of COVID-19 disease. For these organisms, we queried the CROssBAR database to find out the human and viral proteins related to this, which has been shown in yellow nodes and pink nodes. Then we find out all those drugs in CROssBAR which target these proteins, both viral and human. The blue nodes represent the pathways of which these targets are these proteins and genes are a part of. Thank you.